Welcome back to our class. Today we're going to be talking about the prediction for paper 2. So basically it could be a prediction or a scope, but let it be a guideline for you to prepare for your exam. And I know that by the end of this lesson, you must be able to get a distinction. Those who have the distinction material book, I know that the distinction is already in your hands. All right, let's start without wasting time. The first question. The bond that forms between the amino acids during protein synthesis. How do you call that bond? So we call that bond peptide bond. Peptide bond. An organelle, I like to start with the terminologies because this one, it warms you up so that you're able to prepare for the exam or the terminologies of the terminologies of the paper. All right, the organelles that contain DNA, which is used to trace the female ancestry. Remember, we have 3 organelles which contain DNA: nuclear DNA, the nucleus, which you call the nuclear DNA, then you have the choroplastic DNA, so it's found in the chloroplast. Then you have the mitochondrial DNA, which is found in the mitochondrion. But it's only the mitochondrion which is used to trace the female ancestor. Therefore, the answer is going to be mitochondrion. Mitochondrion. Right. The saying undifferentiated cells in an animal that have the potential to become any type of tissue. It means that they can develop into other body cells. So we call them stem cells. A change in the sequence of nitrogenous bases resulting in the formation of a different protein. So such thing, since it is change of the bases, so it's gonna be point. You can call it point. Yeah, because the bases are specific. You can call it point, or we can call it gene. Yes, gene, but because they're saying that change in sudden change in science, we call it a mutation. So in actual sense, maybe the answer could be mutation or gene mutation or point mutation. So yeah, natural shape of DNA. Always they ask you what is the natural shape of DNA. No any other shape. We call it double helix. The permanent disappearance of species, from the earth. So how do you call it? We call it extinction. Yes, the type of variation where there is a range of intermediate types. For example, if you have look at the size, you have big, small, medium, extra big, or extra XXL. Call it XXL. Then you have the small. So a range of phenotypes, but it's one character. So we call it a continuous variation. So when you say that that type, that's the variation, and then they're saying range, so it is continuous. You have different range of this character. Very dark-skinned, light-skinned, very light-skinned. You see, it's a range of character, but what gene is it? Which character is it? That is skin color. Yes, so it becomes intermediate continuous variation. An allele that is expressed in the phenotype in a heterozygous state, meaning that if you look at the heterozygous state, for example, capital T, small t, this allele, it means that if T for tall and small t is for short, so if the individual is like this, capital T, small t, this individual is going to be tall. So how do you call the allele for tallness? So we call it, how do you call it? We call it dominant allele. So it's dominant allele. Yes, we call it dominant allele. The position of an allele on a chromosome. How do you call the position of an allele? Or a position of a gene on a chromosome. You can change it and it's the same thing. So we call it locus. If there are many, call them loci. But since one is called locus. Let's go to other types, and then we see. I know that you have not yet feel like you are ready for these terminologies. So let's have another example. The type of inheritance in which produce intermediate phenotypes, you see. So you produce intermediate phenotypes. So how do you call that kind of dominance? So we call it incomplete dominance. Why do we call it incomplete dominance? For example, you have black crossed with white, and then you produce an intermediate color, which is gray. So how do we call this type of dominance? How do we call this type of inheritance? So we call it incomplete dominance, because neither of the allele is completely dominant over the other. If you look at the previous one, we see that tall crossed with short, and then you get what we call tall, meaning that tall is completely dominant over short. So the point where adjacent chromatids overlap during crossing over, so we call it chiasma. If one is called chiasma, if there are many, we call them chiasmata. The process where information is copied from DNA to messenger RNA, how do you call that? We call it transcription. Formation of the messenger RNA. From DNA, we call it transcription. The sugar found in DNA molecule, how do you call that sugar? Deoxyribose. From the name DNA, which means deoxyribonucleic acid, meaning that it has a sugar called deoxyribose sugar. The bond that occur between nitrogenous bases in the DNA molecule, how do you call those bonds between the nitrogenous bases? We call them hydrogen bonds. So to be more specific, call it weak hydrogen bonds. All right. Manipulation means that you are using the man. That's why you call it manipulation of organisms gene. When you manipulate, actually you are an engineer. Yes. So it means that if you are an engineer, yes, then it means that what are you engineering? You are engineering genes. Therefore, manipulation, engineering genes, how do you call that? To bring the desired characteristics. So the answer is already there. So we're going to call it, since you are manipulating the genes, therefore it's going to be genetic engineering. The representation showing the arrangement of a diploid set of chromosomes. How do you call representation showing the arrangement of diploid set of chromosomes? So if you look at them, meaning that there are sets from chromosome pair number 1 to the chromosome pair number last. So how do you call that? Since you are describing the nucleus, so the nucleus in science is called karyo. But because you are looking at the type, how are they arranged? So it's going to become, which type of nucleus is that? Hence the name karyotype. So we call it karyotype. All right, let's go to match column example, and then we see if you are ready for this paper. Source of variation in the species. Random arrangement, random mating. Remember, random mating, you don't have to be specific that I want the one which looks like this, I want the one who looks like this. Whoever comes, you go with that. Random mating. Random fertilization. How many sperms are being produced in a single ejaculation? Over 4 to 500 million sperms are being released in a single ejaculation, but only one, and you don't know what is that one. So since there are a lot and we don't know which one is gonna do so, it's randomly done and each sperm is different from one another. Therefore, we're gonna call it random fertilization. And this is also a key for bringing variation. That's why your brother might look different from you, because of random fertilization. Therefore, both of them, they contribute to variation. So the answer is both A and B. Organisms possess, an organism possesses two factors which separate so that each gamete contains only one of these factors. How do you call that? Law of inheritance, law of dominance? Not really. Principle of independent assortment. Organisms possess two factors which separate so that each gamete contains only one. Law of principle of independent assortment. Not really. So the answer here is gonna be none. Reproductive isolation mechanism in species. Breeding at the same, this makes it to be wrong. Same season? No. They can't be separated if they are breeding at the same season. Infertile offspring. Infertile offspring. Meaning that you produce like a horse and a donkey, then you produce a male which is infertile. So this looks to be B only. And you get your 2 marks, 2 marks, and 2 marks. Then we go to the next question and we see what is it talking about. So if you look at this section, it's still giving you a dose to see that, yeah, the dose is enough so that you don't have issues with the terminologies. A section of DNA, the DNA molecule that code for a specific characteristic, we call it a gene, is a section of DNA that control a specific characteristic or a particular characteristic or a particular trait. Sometimes we call it trait. Yes. Pattern of black bears resulting from DNA analysis. So how do you call that? They're not looking at the process. They're looking at those patterns you produce. So we call it DNA profile. The process is DNA profiling, but the results is the profile. But whose profile? For DNA. Evolution characterized by long period of change and rapid period of no change. How do you call that? We call it punctuated, yes, equilibrium. Punctuated equilibrium. Yes, punctuated equilibrium. Okay, they're saying that the production of genetically identical copies of organisms, how do you call that? Like you are exactly the same. So you, in science, you call it you are clone. You are a clone. You are a clone. So the process is gonna be cloning. Yes, undifferentiated cells in animal that have a potential to become any type of cell. So we say that we call them stem cells. So you can't remember this, then it means that you have a problem with cerebrum. A genetic disorder that results in a person cannot distinguish between colors. How do you call that genetic disorder? We call it color blindness. Call it color blindness. The part of plant where male gametes are produced. Remember, we call them anthers. Anthers. Yes, I don't want to go to the distinction material book to show each and everything because it will be taking a lot of time, but I think you know what I'm talking about. The structure that joins two chromatids together. How do you call the structure that joins two chromatids together? Central. That structure joins the two chromatids together. The central body, centromere. Okay. Then we are saying that The type of dominancy, neither in which neither of the two allele is dominant over each other, resulting in offspring with an intermediate. Who can tell me the answer? Who can tell me the answer? Who can tell me the answer? Intermediate. Here, the first one, you have the complete dominance, you have the codominance. I think you didn't get it right. The answer is none, because we talked about it before. So that's why I put these questions to check you up, see that you are able to remember. All right, so none of this, because this one brings a mixture of two, and then this one brings the one which is dominant becomes expressed in the phenotype. The point where two chromatids overlap during crossing over, what's the answer? You forgot. The answer is chiasma, and I said if there are many, we call them chiasmata. The scientists who produced the law of segregation, what's the answer? None, because this one, Darwinism, Lamarckism, they are in evolution. All right. So the one who did that is Georg Mendel. All right. The position of a gene. The position of a gene on a chromosome, what did we say? You forgot, is locus. The type of evolution characterized by long period of change and followed by a short period of rapid change. What did we say? How did we say? Punctuated equilibrium. If you forgot, the natural shape of DNA, what did we say? Double helix. If you guys can't remember what we said, we just said double helix. The type of bond formed between two amino acids, what did we say? Peptide bond. The type of vision which is shared by primates that allow deep perception, how do you call that? Is the binocular vision. Yes, or stereoscopic. Right. The type of dominance in which results in the intermediate of phenotype, what did we say? Incomplete dominancy. Yeah, you guys, if you are not able to remember what you have said, then it means that you need to catch up. I have to go to the catch-ups, and we talk about the catch-ups. Then the fluid of the nucleus, where the free-floating nucleotides are found, how do you call that? Nucleoplasm. Entangled mass of chromosomes located within the nucleus. How do you call that? We call them chromatin network. The division of the cytoplasm after nuclear division. How do you call the division of the cytoplasm after nuclear division? When you say division of the cytoplasm, division or splitting, or how do you call that? Does it require energy? All right, yes. So since it's a cytoplasm, so I will call it cytokinesis. The name for X and Y chromosome in the human, we call it gonozome. So guys, that is our first section of the terminologies, and I think guys, you are now well to go, and then kill the paper, because the paper should not kill you. Let's go to the first section of that. But before I go there, don't forget this. Yeah, so your paper 2 will be will be DNA, meiosis, genetics, evolution. And if you look at this, check the marks you're supposed to go for. Yes. All right. So we continue to the next question. So if you look at this question, they are saying that process shown the diagram above. Which process is shown this diagram? So if you look at this process, we shall see that this process is, let me do like this. Which process is that? If you look at that diagram, we see that this is DNA replication, because you have a new strand here, you have a strand here, old strand, and then you have an old strand, and then you have new strands being formed. And because of these weak hydrogen bonds, it means that each molecule is DNA. Therefore, I will say that this process is called DNA replication. Chemical bond between labeled one. If we clear this up, let's find out which chemical bond is labeled one. So these are weak hydrogen bonds. Weak hydrogen bonds. Yes. Then we go the next question, saying that collective name 1 for 2, 3, and 4. 2, 3, 4. So the answer here is gonna be the collective name. They are looking for the collective name. So the collective name is going to be the nucleotide. Full name for nitrogenous base 5. What is 5? 5 is here. Here you have adenine. Automatically this is going to be thymine. So 5 becomes thymine. Okay. Thymine. All right. During interphase, during the phase of cell cycle, does it take place? When does it take place? So basically, this takes place during interphase. Where in the cell does this process takes place? It takes place in the nucleus. So guys, yeah, that is the questions which let's go to the second question. But before we go to the second question, you need to know how to label. Basically, this is DNA, and this process is transcription. So you have messenger RNA being formed. So R is messenger RNA. 3 is the amino acid being brought. Yeah. This is amino acid, and then this is the messenger RNA aligned itself on the ribosome. Remember, sometimes supposed to have 2 bases, the minor subunit and the major subunit. And then you have the anticodons, then you have the codons on the messenger, and then here is the nucleus, and then here all this place is the cytoplasm. So once you know that, now you are able to go and try to answer the questions they have asked you. Okay, so we go back and ask ourselves, identify process Z. Identify process labeled Z. So if we check here now, we are saying that what is Z? Which process is Z? So we say that Z, the process of Z is transcription. Then molecule R, where is molecule R? Is this, which is a messenger RNA. And then we're saying that organelle Q, this organelle, so it's gonna be ribosome. Give the collective name of O. Give the collective name of O. We say that these are anticodon. It's anticodon. Determine the sequence of nitrogenous bases at area S. Area S. They're saying determine the sequence of nitrogenous bases at area S. So when you go back to area S, we shall say that T goes with A, C goes with the G, and then A goes with T. So it's going to be AGT. Why? Because it is DNA versus DNA. It's not messenger. That's why there is no U. Then they're saying that which strand was used as a template. Which strand was used as a template. So if you look at, you have strand 1 and strand 2. So we see that what is formed here, that's strand 1, and then this is strand 2, of which this strand, they say that if you look at it here, we need to check out here. What does this A, because this is messenger, if this is messenger, what does this one correspond with? A goes with the T, and then U goes with A, and then C goes with the G. So I'm going to look for the strand which has this, because this is a mirror for this. Therefore, this became a template. TAG. So it is strand 1. Shows that strand 1 is the one which is used as a template for that case. Then they are saying that which amino acid 3, 4, 5 will be brought by P. Will be brought by P. We go back and find out what is P. So we see a P. I don't know there. Now I need to go back, and then again I only have this and this. I don't have it here. Cool. I will have to go back again, because now this and this is the same, ne? So I have to go back here and I find out, because this is strand, this is strand 1, but still I don't have it here. So it means that to get this, because this and this is the same, to get this I have to know what is this, but to get this I have to know what is this. Therefore I'm gonna say TCA, that is for this. Now I need to find out what is this. So it's gonna be AGT. So this is this. Okay, now once I know this, now I need to find out what is this. So it's gonna be A goes with U, because it is tRNA, CA. You see? So now, since now I know the messenger RNA here, I need to know the transfer RNA. So the UCA will go with AGU. So I have to look for AGU. AGU is here. So amino acid number 4 is the one which is gonna come. Amino acid number 4. Then they're saying that name the type of sugar that forms part of the structure molecule R. Where is structure molecule R, part of sugar? So we said it ribose sugar. Yes. Then we go to the next question of DNA again, and then we see the diagram represents the stage for protein synthesis. Okay, so if you look at this, since you see that this is a ribosome and this is a messenger, then automatically this is a translation. So it is translation. So now, since that is translation, so now it means that identify the organelle 3. We say that organelle 3 is a ribosome. Then describe the role of molecule 1. Describe the role of molecule 1 during the stage of protein synthesis. So basically, molecule 1 brings the required, bring the required amino acid to the ribosome. So it brings the required amino acid to the ribosome, or brings a specific amino acid to the ribosome when its anticodon matches the codon of the messenger. So it brings the required amino acid to the ribosome when its anticodon, or when tRNA anticodon matches the codon of the messenger. So that's how you're supposed to explain that. Give the sequence of nitrogenous bases at number 2. So at number 2. So number 2 is going to be CCU, because this is messenger, it is transfer RNA. Since it is RNA, so it's, A goes with U. Then they're saying that on the DNA molecule that code for histamine, give the sequence of nitrogenous base on DNA. Bases on DNA that code for histamine. So if you look at this, actually it's supposed to be replaced with the T. Okay, sharp. Let's find out. Since this is tRNA, so I'm looking for DNA. So G goes with the C, this goes with the A, and this goes with the U. That is messenger RNA. This is transfer RNA, this is messenger RNA. Now I need to convert messenger RNA to DNA. So it's going to be GTA. GTA. So that's why I said this is replaced with the T. So it means that it's going to be GTA. So I have to convert it into 2 times, because it is, I'm changing from tRNA to messenger RNA to DNA. During the synthesis of the same protein, there was a change in the amino acid sequence, because alanine was replaced with tryptophan. Alanine was replaced with tryptophan. Explain the possible reason for this. So if this happens, so we call this a mutation. So it means that it was due to a mutation. So you mentioning us that it's a mutation, is a change, you might get a mark. So what is other 2 marks? So they say that alanine, this alanine, of which alanine is GGG, was replaced with tryptophan, which is GAA. Yes, that means that during the DNA sequence, changed from GG to GAA. That's the meaning. You understand? So basically it means that the G's were replaced with A's. You see? Let me check properly. Alanine was replaced with tryptophan, meaning that there was alanine was replaced with tryptophan. So that's the meaning that the GG was replaced with the AA, making it to be a different amino acid. So basically you have to tell us what really happened. If you see that alanine is coded by GG, and the tryptophan is coded by GAA, then it means that the G and G was replaced with A and A, and then because now on DNA molecule, therefore resulted in different amino acid. And this process of changes is called mutation. That's why your first mark at least is going to be going to the mutation, and then you tell us what exactly happened, that GG was replaced with AA, which resulted in the formation of a different amino acid. So there is no way you're gonna skip a question concerning about mutation. So just know that a question for mutation is gonna come. So please, when it comes, make sure that you answer that question very well. Then we go to the next question. What is X? Is a amino acid. What is Y? Y is 3 bases. So I will call these ones anticodons. And then Z, if you look at Z is going to be the messenger RNA. So which process is this? Basically, this is a translation process. So they're saying that identify molecule X. So if you look at it, molecule X, we said molecule X is amino acid. Z, we said Z is a messenger RNA. Give the nitrogenous base sequence of DNA triplet complementary to the middle codon on Z. So if you look at this, it's messenger. They say the middle, the triplet which code for the middle. Middle is so it's gonna be on DNA. So it's gonna be A goes with the T, U goes with the A, and then G goes with C. So it's gonna be TAC, which means that TAC. Nitrogenous base sequence of Y. Where is Y? So in this case, since I have this CAU, so I'm gonna convert into transferrin A, which is gonna be G, GU, and then A. So that is going to be the answer for Y. Name the process and describe the process. Name and describe the process shown in the diagram above. So in this case, we want you to explain translation. So if you go our distinction material book, it is clearly stated there, or according to the messenger RNA codon, the transferrin A with the complementary anticodon will bring the required amino acid to the ribosome. The amino acids are joined together by peptide bond to form a required protein. So that's what you support. You can't go to the paper without knowing the transcription, the translation, the DNA replication. You can't do that. If you do that, just know that you are messing yourself up. All right, so we got the next question. So the table below shows the codon, shows the codons that code for some amino acids. A mutation caused by DNA base triplet to change from CTG to CTA. Describe the effect of this mutation. So if there is CTG, which code for triplet on DNA, what do we have here? We have messenger. So if it is CTG, then it's gonna be CTA. I need to first convert this to because this is DNA, and what I have here is messenger, so I have to convert into messenger RNA. C goes with G, and then T goes with A, and then C, and then this one I also have to convert it, so it's gonna be GAU. So what does this code for? GAC. GAC is aspartic acid. So this is aspartic acid. And what is GAU? GAU is also aspartic acid. Even this one is aspartic acid. So it means that there is no change in the protein. Therefore, I have to tell that the codon the codon GAC, yes, changed on the messenger RNA, changed to GAU. You understand? Both they code for the same amino acid, aspartic acid. Therefore, there was no change in the protein, or there was no effect on the protein which is formed. Yeah, so in that case, you have to first identify the sequence which is being given here. Is it DNA or messenger RNA or transfer RNA? And then what is given here, is it DNA, messenger RNA, transfer RNA? If it is the same, you keep the codes. If it's different, then you have to convert so that they are the same. So in this case, what is being given is DNA, and what was given here is messenger. Therefore, I had to convert from DNA to messenger so that I'm able to match them, and then I read them. If I find out that the amino acids are the same, coded by these codes, therefore there is no change in the protein. But if I find out that this code is for aspartic acid and then this code is for leucine, for example, then it means that there will be a difference in the amino acid. Therefore, there will be a change in the protein. Then we go the next question, tabulate difference between DNA and RNA nucleotide. Here they are talking about nucleotide, but if they ask you to tabulate, make sure that you draw a table. The moment you draw a table, that's 1 mark. And if you draw a table, don't just draw a table which is just empty like T like this, 0. So make sure that DNA, it must be complete, nucleotide, RNA, nucleotide, then we'll give you a mark there, that's 1 mark. Then point here, DNA has ribose sugar, DNA has deoxyribose sugar, RNA has ribose sugar, you get a mark. DNA, you tell me has thymine base, RNA has, what does it have? Has uracil base. Then you get another mark. You get 1 mark here, you get 1 mark here, you get 1 mark here. That's all. Make sure that whenever they ask you to tabulate, don't just draw a table, which some of you just draw because you want to finish and leave, which is wrong. You are messing up your life. Yes, someone was saying that the warmer the blanket, the colder the future. Hey, I liked that statement when I was busy online. I like that statement. The warmer the blanket, the colder the future. Yes, that's it. So if you like the blanket too much, just know that the future is going to be colder. And if you leave the blanket so it will become cold, then it means that the future is going to be warmer. So that is it. The next question is about the DNA profiling. Name the technique which is being used. Is DNA profiling. Then they're asking you which suspect 1, 2 was at crime scene. If you look at here, sample, suspect 1, suspect 2. This is the sample from the victim, and then this is a sample from the crime scene. So it means that we have to match. So if you look at this, matches with this matches with this, all of this match with this. So automatically suspect 1 was at the crime scene. Give one visible reason for that. If you look at it, the DNA profile, you have to, don't just call them DNA, DNA profile or DNA bands. Please don't change the words. Bands of suspect 1, yes, is identical to the DNA profile in the DNA profile of the crime scene. You see, DNA profile of suspect 1, yes, is identical or is the same as that of the crime scene. So that is the reason why we gave that to be the answer. Then they're saying that give 2 possible reasons why the suspect identified question might not be convicted if the evidence is used. First of all, yes, we know that DNA is unique, but it might be manipulated. What if someone went there and then dropped it? There could be human error. The suspect can say that, no, that is human error. It could be contaminated with something else. You understand? Yes, maybe they used the too small amount of, what did they use? Blood, what? Small sample. They used a small sample from the crime scene. Can say that, no, that's not mine. You understand? Yes. So that's some of the reasons that might not be convicted. Then they are saying that explain why the technique mentioned above is used in the paternity test. Explain how. They're asking you how this technique is being used in the paternity test. So basically, if someone is claiming that this is my child, you have to first identify the DNA profile of the child and the DNA profile of the mother. You understand? The DNA profile of the child and DNA profile of the mother. So you remove all the DNA, the bands which match with the child and the mother. The remaining bands of the child must match with the father, the possible father. And if they match, therefore that will be the father. But if they don't match, then that suspected father won't be the father, because they don't match. So DNA profile of the mother and the child Number one. Number two, match all the DNA profile of the child with the mother. Once that's done, now remove the remaining profile must match with that of the what? Must match with that of the father. If they match with that suspected father, then automatically that is the father. If it does not match, then it means that that is not the father. So now from here we're gonna go to what we call our catch-ups, and then we see some of the questions in the catch-ups. So if we go to our catch-up, we see the question describe the structure of DNA. So in this case, you have to describe how DNA is structured. Remember, we say that DNA is made up of nucleotide. Yes, made up of what? Nucleotide. Nucleotide is made up of phosphate, deoxyribose sugar, and nitrogenous bases. Then you tell us adenine, thymine, guanine, cytosine, whereby adenine pairs with thymine and guanine pairs with cytosine. And then you tell us, you just basically describe the structure of DNA in that regard. It has two strands joined together by weak hydrogen bonds. So you can't go to the paper without knowing how to do the proper description for this structure. So if you go back here at the beginning of DNA, you just look at the structure of DNA and then you are able to describe that structure based on what you already see. You just have to know that that's the structure, and then you have to describe the structure. So now we got the next question. They are saying, what's the next question? What are the monomers? Of DNA? We said they are nucleotides. Who discovered the structure of DNA? That's James Watson, and then Francis Crick. If you look at the people who are involved, then automatically you have to also include Maurice Wilkin, and then Rosalind Franklin. So those are the four people who are involved. Who discovered the structure of DNA using X-ray diffraction picture? That is Maurice Wilkin, and Rosalind Franklin. Then they are saying that in which year was that? Was 19. Basically, when was the Nobel Prize being given? So that was being discovered in 1962. So other than nuclear DNA, name other types of DNA. We have what we call the mitochondrial DNA, the one which is found in the mitochondria, and then we have what we call the chloroplastic DNA. What is DNA? Replication is the process whereby DNA makes exact copy of itself. When does it occur? It occurs during interphase. Where does it occur? It occurs in the nucleus. Why does it occur? To increase the chromosome number. So basically here you look at the distinction material book, and then you see that all these things we are talking about, they are there. How does it occur? Here you describe the structure of you describe the process of DNA replication. So that's what you're supposed to describe. So for example, in this case, what you have been talking about is here. What is DNA replication? When does it occur? Where does it occur? Why does it occur? You see, double the chromosome number. And then how does it occur? You X-ray DNA replication. Double helix DNA unwinds weak hydrogen bond break to form two separate strands. Each is acting as a template using free floating DNA nucleotides. Remember, in transcription we used free floating RNA nucleotide. Here we use free floating DNA nucleotide from the nucleoplasm. A complementary strand is formed whereby A pairs with T, and then G pairs with C. Two new DNA molecules are formed. These DNA, they are genetically, they might not be, they swap the strands, but if you look at them genetically, they're identical. So you're saying that is identical and someone else saying that genetically identical, those are two different stories. With one original strand and one new strand, this process is controlled by the enzyme. So you get the marks for that question, which asks you to describe how does it occur. What are some of the functions of DNA profiling? This one is a simple thing. I don't usually go through them, but we can use it for paternity test, identification of individuals at the crime, the crime or crime identification. You can talk about diagnosis of inherited disorders. You can talk about personal identification or relatives, or for example, when someone is dead or burnt, so we are able to identify that individual. So that is DNA profiling. What are disadvantages? Sometimes you call them debates about, or what are the debates about DNA profiling? Very expensive. Number two, sometimes a person they can they cannot, it might not be 100% correct due to the human errors. You understand? Human errors. Not all hospitals have the necessary equipments, can also lead to the fault results. You see? Yeah, sometimes you can also frame the individual at the scene. Someone did it and then dropped something which belongs to you, you see. So all this can be a reason. What is the function of messenger RNA? It carries coded information from DNA to cytoplasm so that a protein can be formed. So it basically carries coded information from DNA, that is from the nucleus to the cytoplasm, so that a protein can be formed. Why is the messenger RNA has a specific sequence? Or basically, what's the function of the messenger RNA? Sorry, why is the messenger RNA has a specific sequence? This one is basically to determine the specific sequence of these amino acids, hence a specific protein. So the specific sequence of messenger will determine the specific sequence of transfer with its amino acids, and then it will determine the specific protein. Then we go, what is protein synthesis? We have talked about all this, but we are just doing a recap or a catch. Then what is DNA replication? What is protein synthesis? Is the process whereby DNA makes exact copy of, is the process whereby the cell makes proteins. The process where proteins are being made in the cell, process where the cell makes proteins. So this process is divided into two, that is transcription and translation. Okay. And then transcription is the formation of messenger, and translation is the formation of polypeptide chain. Which organelle does transcription take place? Organelle occurs in the nucleus. The organelle is nucleus. That is where transcription takes place. On which organelle does translation takes place? It occurs on the ribosome. Describe the process of transcription. Describe the process of transcription. Here we talk about double helix DNA unwinds, weak hydrogen bonds break to form two separate strands, one acting as a template for formation of messenger RNA using free-floating RNA nucleotide from the nucleoplasm. A complementary strand is formed whereby adenine goes with the pairs with uracil and cytosine with guanine. Now messenger RNA has a coded message for protein synthesis, which moves out of the nucleus through the nuclear pore to the ribosome. You see, that is transcription. That is transcription. So you have to know the process of transcription. And then what is translation? According to the messenger RNA codon, the transfer RNA with the complementary anticodon bring the required amino acid to the ribosome. The amino acids are joined together by peptide bond to form a required protein. So equivalent is a key point. So you guys who are using distinction material book, I think now you know what you're supposed to do. Please use it effectively so that you're able to succeed effectively. What is the difference between DNA nucleotide and RNA nucleotide? When talk about DNA nucleotide, nucleotide of DNA, we shall see that this nucleotide, they are not asking about the strand. It has thymine for DNA, for RNA it has uracil. It has deoxyribose for DNA, for RNA has ribose. Yes. So that is it. What is the difference between the DNA replication and transcription? Here you look at what happens in DNA replication but does not happen in transcription. Here you form two new DNA formed. Here you form a protein at the end. Here we see that we use free floating DNA nucleotide. Here we use free floating RNA nucleotide. This one occurs in the nucleus only. This one occurs. Oh, they say transcription, not protein synthesis. So it means that here occurs in the nucleus. Also, this one occurs in the nucleus. Oh, yeah. So at the end of the day, here you have formed two new DNA. Here you form messenger RNA. Here you use adenine goes with thymine. Here adenine goes with uracil. So basically there are a lot of differences, even similarities. Here we see that we have DNA double helix DNA unwind. Even here double helix DNA unwind. Weak hydrogen bonds break. Here even weak hydrogen bonds break. This reaction occurs, or this process occurs in the nucleus. This process occurs in the nucleus. So you see that there are similarities which are happening between them. So from there we have to go to what we call meiosis. But before we go to the catch-up of meiosis, we have to go back and see where we stopped with the meiosis. In this case, we're going to start with what we call the karyotype. So this karyotype, if you look at it, we see that the karyotype, does it have any abnormalities? It doesn't have any abnormalities. Oh, it has here two of them, but the others are single. Okay, name the type of mutation that occurred during the production of this gamete. So it shows that the type of mutation is chromosomal mutation. Chromosomal mutation. What type of gamete is represented by the karyotype? Because of X and Y on 23, so it means that it's gonna be a male. In this gamete, give the number of autosomes. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. So there are 22. There's no one which is mutation where there are two of them. So there are 22. Gonosomes, it's 1, 2. So there are 2. They're saying that the gamete, remember gonosomes are the sex chromosome. The gamete fuses with the normal gamete. Gamete fuses with the normal gamete. Using X and Y representation. Write the sex chromosome of the offspring. sex chromosome of the offspring. Now the question here, the challenge here is that they did not specify which normal gamete. Oh, normal. It means that if it is fusion, then it means that it fertilized the normal gamete, which is the female. Yeah, so there's no challenge. So since this is X and Y, so it's going to be X and Y, yes, then plus a normal gamete, which is going to be, remember the normal individual is going to be like this for the female, only one comes. So it means that this individual at the last is going to be XX then Y, XX then Y. So that will be the phenotype of the genotype of that individual. Then we go to question number 4. The diagram will represent the cell in the early stage of meiosis. So here we see that this is a cell nuclear membrane breaking. Then this is cell membrane. This is nucleoplasm. Then they're asking the phase of meiosis represented. The phase of meiosis represented. So if you look at this, you see that crossing over is going to take place soon. So it means that this is prophase 1. Name the chromatid shown. Number of chromatid. Number of chromatid is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So basically the number of chromatids are 12 because each chromosome has 2 chromatids. It has this and has this. So number of homologous chromosome pair, if you look at them, this 1, this 2, this 3. So the answer is going to be 3 chromosome pair. Then they are saying that identify structure. R, we say the nuclear membrane. S, we say that is cell membrane. And then T, we said is the nucleoplasm, the fluid of the nucleus. Name 2 organelles in the animal where meiosis takes place. 2 organs in the animal where meiosis takes place. It occurs, they say the animal in individuals. So it takes in the testis, yes, and also it takes place at the ovaries. Yes, those are the 2 places where these individuals, where this can take place. Then from there, we go back to our catch-up and then we see what is on that side of our catch-up. There are a lot of questions you can do, yes, but crossing over, you have to know the process of crossing over, you see, yes. So let's go back to our catch-up and then we see. So this is meiosis. I hope you guys, I said that this mutation will be coming. So make sure that you prepare that mutation properly. So when you go back to our catch-up of meiosis, give 2 importance of meiosis. Meiosis, first of all, the importance of meiosis is very good for variation. It's very good for variation. Number 2, production of haploid gametes. Number 3, those things are here in the distinction material book. I don't want to go there, you understand. The doubling effect, solving the problem of the what, the problem of what, the doubling effect solves the problem of the halving effect of meiosis solve the problem of doubling effect. Then you also, we said variation, gametes, and also maintaining the chromosome number from generation to generation. That's the meaning that the halving effect of meiosis will solve the problem of doubling effect of during fertilization. Yes. So importance of meiosis you have to know. In which structure does meiosis take place in the plant cell and animal cell? We say that here in the anthers, and then also in the ovaries. And in animal cell, we have already answered it. We say that in the testis, and then the ovaries. Anthers produce pollen grains. Ovaries produce ovules. in plant. Testes produce sperms. Ovaries produce ova. Yes. Then we go the next question. Describe the structure of the karyotype. Here you have to describe what you see. For example, here we see this karyotype. What are you going to describe when it comes to this karyotype? First of all, human karyotype, if you look at it, human karyotype is made up of 46 chromosomes arranged in pairs, each with different from each other by shape, size, and genetic composition. From pair 1 to 12 is called orosomes. The 23rd pair is called gonosome. The gonosomes consist of X and Y, whereby X and X becomes a female, X and Y becomes a male. X is longer than female. So basically you're describing what is inside, which type of nucleus does it have. Yes. Then describe the process of crossing over. Here in the process of crossing over, you describe that homologous chromosomes, they align together. Homologous chromosomes, they align together. One chromatid of adjacent chromosome overlap. One chromatid of adjacent chromosome, or one chromatid of each adjacent. Add the word adjacent here. Adjacent chromosome overlap with the chromatid of the homologous pair. You understand? It must be adjacent, this and this. This cannot cross with this. It must be adjacent from the homologous chromosomes. Overlap with another homologous pair. This process is called crossing over, and it takes place at a point called chiasma. The chromosomes separate in such a way that each has new genetic material from both parents. So the process of crossing over, that is it. You have to be knowing it by heart. Draw the diagram of the cell after metaphase with the originally with the 4 chromosomes. They don't usually bring you this question like this. Sometimes we just bring to say that draw this cell at the end of telophase 1. At the end of telophase 1, then it means that you will be able to, the caption, don't forget to label telophase 1. Remember that now you're going to have 2 chromosomes, from 1 chromosome from there, 1 chromosome from there, 1 chromosome from there, 1 2 3 4 5 6, yeah, and then another chromosome from there, like that, you see. And then the shadings must be right, because here crossing over is going to take place. So you show us at the end, make sure that you label cell membrane, nuclear membrane, chromosome, centromere, chromatid. So you have to label. Don't draw, even if they don't ask you to draw, make sure that you draw and label. At least you'll get, if the other things are wrong, at least you'll get the 2 marks for labeling. What is the function of crossing over? It brings about genetic variation, which increases the chances. Yes. What's the function? Brings about genetic variation. What is the importance of crossing over? Basically, since we say that it brings about genetic variation, this helps the organisms to survive differently in the environment. Yes. If you are different from me, it means that you will be doing things I can't do. So we're going to survive differently in the environment. Meaning that the environment, different environment will treat us differently. That's the meaning. Other than crossing over, what is the other cause of variation in meiosis? Other than crossing over, what is the other cause of variation? So you can talk about random arrangement of chromosomes during metaphase, random fertilization. They say during meiosis. So random arrangement of chromosomes during metaphase, and also mutation because also occurs during meiosis. It can occur during meiosis, like the chromosome mutation. So those are the 2 points, because they said meiosis. So if it is out of meiosis, then you can talk about random fertilization, random mating, you see. Yes. What is non-disjunction and how does it lead to Down syndrome? What is non-disjunction? This is the failure of the chromosomes to separate during anaphase. It can be anaphase 1 or it can be anaphase 2. Give two difference between meiosis 1 and meiosis 2. We see that here when it comes to this, I've not explained how it leads to Down syndrome. So in this case, you have to tell us that non-disjunction occurs at a process at a metaphase, and then non-metaphase, anaphase. Let me explain to you nicely here when so that I don't waste too much time. Okay. With non-disjunction, yes. So if you look at non-disjunction here, we see that here you have in metaphase they were fine. You see, they were fine like this, but in anaphase, this is anaphase 1, we see that this is supposed to separate to go to one cell, this is supposed to go to one cell. So what happens? They separated properly in anaphase 1. In anaphase 2, the chromosome is supposed to split, you see, supposed to split. But now, unfortunately, this one split properly, it went this side, and this one went this side. That's why you see this one here, and that's why you see this one here. But the second one, the whole of it was put one side. That's why you see 2 of them here, you see. So it means that the cell, instead of having 2 chromosomes, because this one is supposed to be here, it will have 2 plus 1. Now instead of having 2 chromosomes, it will have, because this other one went to this side, it will have 1 instead of 2. So these ones where the separation happened properly, it had 2 of them, you see. So if the cell has 23 chromosomes, then it means that now this cell is going to have 23 plus 1. That's why I say n plus 1. So it's going to be 24. And then if this cell is supposed to have 23 chromosomes, then it's going to have 23 minus 1, because the other one went to here. So it's going to be 22. And then this one, it's normal, it's going to be 23, it's going to be 23. So now, if this chromosome fertilizes a normal chromosome, this cell fertilizes normal cell, so you're going to see that 24 plus 23 is going to be 24 plus 23, so it's going to give me 47. But yet it's supposed to be 46, because it's supposed to be 23 plus 23. So this condition, if it happens on chromosome number 21, therefore it becomes trisomy 21. You have 3 chromosomes of 21 in that individual. Yes, 2 from this cell and then 1 from the normal gamete. So 3 of them. So trisomy 21. And that condition, we describe it as Down syndrome. So that's why it's saying that each nucleus should contain 23 chromosomes. After meiosis, if non-disjunction occurs, one nucleus will contain 22, while the other will contain 24. You see? Yes, that's what I've been explaining. When either of these fuse with the normal gamete, automatically, if this one fuses with the normal gamete 23, it's going to be 45. If this one fuses with the normal gamete, it's going to be 47 instead of 46. And this, if it happens on chromosome number 21, therefore is going to be called trisomy 21 or Down syndrome. Yes, so this is very common in people who are aging up. Then the difference between meiosis 1 and meiosis 2. Meiosis 1, you look at phase by phase. Chromosome arrange themselves at the equator in homologous pair, that is metaphase. Chromosome line along the equator individually, that's metaphase 2. You see, the whole chromosome moved opposite pole, that is anaphase. The daughter chromosomes or chromatids move to opposite pole, that is anaphase 2. You see, 2 cells are formed at the end of meiosis, that is telophase 2. 4 cells are formed, telophase 1, and then 4 cells are formed, so that is telophase 2. You see, chromosome number is halved, that is 1 nuclear division. Chromosome number is maintained. Here the nucleus does not divide the number of chromosomes, because it is the chromosomes themselves to separate into chromatids. Here crossing over takes place, that is prophase 1, and here there is no crossing over, that is prophase 2. So look at what happens in prophase, what happens in metaphase, what happens in anaphase, and what happens in telophase. Once you know that, then you are able to understand or to get all these answers correct. Then difference between meiosis and mitosis, we say that when you talk about difference between mitosis result in 4 daughter cells. Mitosis result in 2. Meiosis result in the maintaining of the leads to variation. In mitosis there is no variation. In meiosis we see that we have 2 nuclear division. In mitosis you have 1 nuclear division. So you have to know such questions. All right, so we are done with that. Let's go to genetics. Let's go to the questions of genetics. In this plant, it carries edible fruit. Scientists have been studying the inheritance of 2 genes, one for stem texture and another is fruit shape. So because of 2 characters being considered, therefore this is a dihybrid cross. The stem can be smooth, capital N, or prickly, small n, while the fruit shape can be round or elongated. Name the type of cross which is being studied in this characteristic. With this characteristic, so basically it is a dihybrid cross. The hybrid cross. State the dominant characteristic for the stem texture. Dominant characteristic for stem texture. So we see that texture, we have smooth and prickly. So in this case is smooth, so the answer is going to be smooth. Then recessive for fruit shape, which one is small letter? The one which has small letter is the one which is elongated, is the one which is recessive. Then the genotype of plants with prickly stem and elongated. Prickly, remember that prickly is small n, it can only be homozygous recessive, and then elongated with small r. So since now it is a genotype, so I have to combine them together, NNRR, like that. ORRNN. The phenotype of the plant with both genotype NNRR. Genotype NNRR. What is the phenotype? Because of capital N means smooth, and capital R means round. So it's gonna be smooth and round fruit. Smooth stems, smooth what? You have to be specific. Smooth stems, and then round what? Round shape, round fruit. So yeah, that's it. Then we go to the pedigree. How many generation? 1, 2, 3. This means that there is interbreeding. This means these are kids. Yes. Okay. Since now we know that, let's find out. Okay. The question is saying that, the first question is saying that, how many generations are represented in diagram? So we saw that we are 3 generation, 2, 3. They're saying that, give the genotype of 9. So the one who is a female with the cysts fibrosis. So, but they say that they want the genotype. In terms of genotype, is cystic dominant or recessive? They said it's caused by a recessive allele. Therefore, it's gonna be small b, small b. Yes, small b, small b. So this one's gonna be small b, small b. Then they're saying that explain why both individual 5 and 6 must be heterozygous for cystic fibrosis. So if you look at individual what and what, 5 and 6, 5 and 6, yeah, because they produce kids who are homozygous recessive. So if this one is capital B, capital B, capital B, capital B, there is no way how they're gonna produce this. Therefore, they have to be, if one is capital B, small b, where's the second B gonna come from? So there is no way. So they have to be homozygous, heterozygous. Why? So that one small b, capital B, small b, capital B, small b, so that if this and this they combine, they can form that individual, or they can form that individual. Yes. So in this case, you can say that both parent A and B have they are dominant. They have both dominant and recessive alleles. Both parents A and B, yes, they have dominant and recessive allele. Since they do not have, why do they have both recessive and dominant allele? Because they are normal. Is it clear? But their children 8 and 9, yes, they have cystic fibrosis, which means that these kids, they have homozygous recessive alleles. They received one recessive allele from each parent. Ne? So basically, you need to explain that both parents, 5 and 6, they are heterozygous. You understand? Heterozygous, meaning that they have a dominant and a recessive allele. And their kids, 8 and 9, they have cystic fibrosis, which means that they are homozygous recessive. They received or they inherited one recessive allele. From each parent, meaning that one from this parent and one from that parent. Hence, they had to be with that condition. Using the letters on the diagram show the inheritance of cystic fibrosis when heterozygous parents are crossed. Here they are talking about homozygous parents. If it is homozygous parents, it means that you're supposed to cross both of them homozygous, and they have not talked heterozygous. They have not talked about which parents are those. So it means that I can't use P2 F2. So I'm gonna say P1 F1. Then you tell me that phenotype. You don't forget this phenotype. Then the last one is phenotype. Then that were male, normal male without cystic fibrosis, crossed with female without cystic fibrosis, female normal without cystic fibrosis. Don't want to write all that here, but you must write, for example. Then they say that genotype, we go to the genotype. I say, what's the genotype? Both are heterozygous. They said both are heterozygous. This word is very crucial. They say the parents. Why do you say both? Because parents. Then you go to meiosis. They must split, capital B, small b. They must split, capital B, small b. These are gametes. I always dress them up. Gametes. Dress them up. Then from there, fertilization takes place. You can draw a Punnett square or you can cross. So I always tell people, draw a bullet. So this with this, you join the bullets. Capital B, this with that, capital B, small b. Now this with that, you start with the capital letter, capital B, small b. Then that with that is small b, small b. Then these are offsprings. We said F1 offsprings. Yeah. So what is the question? Use A and B to show the inheritance of cyst fibrosis. So in this case, that is it. I can say that the phenotype, we have 3 people without cyst fibrosis, 3 people without cyst fibrosis, and then 1 person with the cyst fibrosis. That's all. That's all. Then the same question, we can look at this question now, and then you see that it's the same story. You can do a genetic cross, and then you see that you get similar answers. The question is saying that the diagram below shows the inheritance of TAR, a rare disease which leads to the destruction of a neuron. It is inherited as autosomal disorder controlled by the allele TA. Okay, sharp. Describe what is meant by autosomal disorder. It's a disorder which is controlled by allele located on autosomal chromosome. Remember, we have the 6 chromosome, and then we have the autosomal chromosome. So it's the disorder which is controlled by alleles found on autosomal chromosomes. Autosomal chromosomes is pair 1 to pair number 22. The pair 23 is the gonosome, which is a sex. How many sons do individual 1 and 2 have? Individual 1 and 2 have how many sons? They're saying sons. It's only this, because this one came and he married the daughter. So the answer there is gonna be 1. Using the individual 3, 4, 7, explain why it could be concluded that TAR disease is controlled by a recessive allele. Why should we individual 1, individual 3, 4, and 7? 3, 4, and 7. Why individual 3, 4, and 7 is, why do we conclude that it's controlled by a recessive allele? So basically, when they ask you such question, you look at the babies, the individuals they have produced. Yes, you look at the individuals they have produced. So in this case, we see that normal, but they produce someone who has the disorder. Let me check and see. Yes, normal people, they produce, it means that this one was heterozygous, and also this one was, it's like the other question, but they are just asking it in a different version. And then they produce kids who are heterozygous, but in this case recessive. Not heterozygous, homozygous recessive. All right, so it means that this one individual inherited 1 recessive allele from 3 and 1 recessive allele from 4. So individual 3 and 4 are both without the disorder. Yes, but they produce a kid number 7 who is having the disorder. So in this case, we see that if they have the disorder, this and this, so as we have seen that individual 3 and 4, they are both without the disorder. So since they don't have a disorder, so it means that these 2 individuals who don't have the disorder, they produce a kid who has the disorder. It means that this individual number 7 received 1 recessive allele from each of them. Which means that this condition is caused by a recessive allele. Then we go to the next question. We are saying that individual 1 and 2 can produce children with 3 possible genotypes. Individual 1 and 2, they can produce kids with the 3. Okay, then they're asking you, List all the genotypes that have 25% chance of being produced. List all that have 25% chance. Or individual 3 and 4. So if you look at 3, if it is capital T, small t, capital T, small t, this and this is going to be capital T, this and this is going to be capital T, small t, this and this is going to be capital T, small t, this and this is going to be small t, small t. So this is 25%, 50. Is it 25%? No, it's 4, 25, 25, 25. So since this is, these two is 50 because it is the same. So it's gonna be 1 out of 4 times 100. So yeah, it's 25. And then this is gonna be 2 out of 4 times 100, which is gonna be 50. And then 1 out of 4 times 100, which is 25. So it means that these two are going to be 25. So the answer here has to be capital T, small t. No. Capital T, and then small t, small t. The other one, which is capital T, small t, is 50%. Is 50%, while this one is 25%. Question number 6, 2.6. They're asking you, in human, hemophilia is caused by a recessive allele, small x. A woman with heterozygous for hemophilia marries a man who is hemophilic. Use the genetic cross to show. If you look at this, they are saying that in human, it's the same story. In human, hemophilia is caused by a recessive allele, a woman who is heterozygous. It means that it's the same as the other one, but now you're using a carrier. So it's going to be P1, F1, for example, phenotype. Phenotype is going to be a woman, heterozygous, normal, from hemophilia crossed with a man who is hemophilic, meaning that hemophilic male. So genotype is gonna be a woman is like this, but they say normal. So capital H, small h, they said heterozygous. And then a male is gonna be hemophilic. Male is like that, so small h there. Then meiosis has to take place so that you split, and then you're gonna have a capital X, capital H, then X, small h. These are gametes. So I'm gonna call these gametes, and then this one is gonna be small h, and then Y. These are gametes. So now from there, I'm gonna have to say P F2, so that now this 1, 2, 3, 4, this with this, so it's gonna be capital H, small h. Then now this with this, it's the same thing. It's gonna be capital H, small h. Then this with this, so it's gonna be S, capital H, small h, and then this with this, so it's gonna be X, small h, and then Y. So basically that's it. That's offsprings. And then they say that use the cross to show the percentage chance of couple having a daughter who is homozygous normal for blood clotting. Homozygous, a daughter who is homozygous normal. Homozygous normal, the chance is 0. There is no homozygous normal here. The chance is 0 because this is normal female heterozygous, hemophilic normal heterozygous. There is no way you're gonna produce a female who is normal unless this person was normal. Yes, so the chance is 0%. So it's 0 out of 4 times 0 out of 2 times 100, which gives me 0% chance. Then we go to the next question. They're saying that is about insecticide. But before we go to this genetic, is it genetic engineering? Yes, so let's do it. Corn are crops that have genetically modified to be insect resistant. The table below shows the percentage of using. Okay. Basically, this is an investigative question. Describe how genetic engineering of BT was done. What did they do? Basically, what they do is the gene for the insect resistance is isolated from another species and then inserted into the DNA of the corn, which is then where it is expressed. So basically, they are doing insertion. They do insertion. So they just get the gene for resistance, then they insert it in the corn. And then once it's done, then that corn is going to be able to resist the disease. So if you, for example, you want something which produce milk, and then this one is resistant to disease. So it can produce milk, but it's not resistant to disease. This one can't produce milk, but is resistant. So I'm gonna go here, get the gene for resistance, then I put it in this cow, which can produce milk. Then it means that the resultant I produce is gonna be producing milk and then resistant to the disease. That's what it happens with genetic engineering. Describe the relationship of land use of BT corn, amount of BT corn and the amount of insecticide used. So we say that as the land use, the land used to grow BT corn increase, the amount of insecticide used decrease. You see, the land use for BT corn, you see, as it increases, the amount of insecticide used also decreases. That's the meaning. I say just describe. They don't want to give you. We don't want the explanation. We don't want the reasons behind that. No. They're saying that state other two plant characteristics that are genetically engineered to increase the food security. State two other characteristics. So if you go back to our genetic engineering, we see that long life, drought resistance, nutrients, increased size, crop yield, herbicide resistance, all this. If we go to our distinction material book, we will find all this in there. We will find all that in there. Yes, genetic engineering. This is what they are talking about. Yes, you see, this is what they are talking about. Crop yield, increase the crop size, pest resistance. This is what they are talking about. Then they are saying that draw a bar graph. Here you can draw a bar graph, but you guys please take note of this. Always students fail when they ask them to draw a bar graph or a line graph or what. The graph to represent the percentage of land use to grow BT coin between 1995 and 2010. You see, 1995 to 2010. So they say that BT coin, meaning that they want this. So it means that you draw remember, bar graph has spaces in between. It has spaces in between. And then this is gonna be X and this is gonna be Y, dependent. So it means that I'm gonna put it there as 1995, 2000, 2005, 2010, and then here I'm gonna put this percentage. Land use grow BT corn in brackets percentage. You have to put the units. and then you put the caption. Then after that, you'll be able to mark and get all the marks. Make sure that at least you know how to do a pie chart, a line graph. I don't think they can bring the histogram because they brought it in the previous paper. Describe the process of speciation. So now before we go to speciation, we have to do the catch up, and then we see what is the catch up all about in this regard. All right, so it's time for catch up. So differentiate between recessive, dominant, phenotype, genotype, and complete dominance, incomplete. So recessive is allele which can only express in a homozygous state in absence of a dominant allele. Is the allele which can only express in the phenotype in the absence of a dominant allele. And then dominant allele is the allele which expresses itself in a heterozygous state. If you have something like this, this one becomes dominant allele. So it's the character which is being expressed in a heterozygous state. A phenotype is a physical description of an organism, while genotype is the genetic description of an organism. Complete dominancy, here it means that if you have capital T, small t, that this is completely dominant over this. So if I cross, this is gonna be this. So it's gonna be, this person is gonna be taller. However much you have a recessive one. So it's gonna be complete dominance is the one where the dominant allele is completely dominant over one allele is completely dominant over the other. Then incomplete is neither of the allele is completely dominant over the other and results in the production of an intermediate organism. So what is a locus? Locus is the position of a gene along a chromosome. And then Mendel's law of segregation, this one describes the alleles separating. Yeah, that basically it states that the alleles for each gene separate during gamete formation. The allele for each gene separate during gamete formation. So that's why you do like this, capital T, so small t, and then you separate them. Then this becomes a gamete. That's what we have been doing. They're saying that disorder which results in the failure of the blood to clot, referred to as hemophilia. Hemophilia. What is sex-linked character? Is a character whose genes are carried on X chromosome or sex chromosomes. They are carried on top of X chromosome. What is sex-linked gene? Is an example. What is sex-linked gene? Is a gene which are carried on X chromosome. An example of a sex-linked disorder, you can talk about hemophilia, and then you can also talk about color blindness. How many allele control blood group? There are 3. And then what are they? Is allele for A, allele for B, and allele for the small allele. How many genes control blood group? It's 1 gene. What is that? Blood group gene. A man who is blood group A marries a woman who is blood group B. Yes. What is the percentage chance of producing blood group O? Yes, you have to do the genetic cross now here, P1, F1, but you have a man of blood group A. The percentage chance is gonna be, I think, 25%. How? Let's just do it a short explanation. But you have to do the whole process the same way a genetic cross. So if you have blood group A, let me just show you here from our distinction material book so that I don't take too much time. Blood group is above. Blood group is above. Yes. So it shows that, yes, it's here. So if you are blood group A, yes, blood group A, and you're heterozygous, and blood group B, you're heterozygous, you have chances of producing blood group, or if this recessive allele and this recessive allele meet, you can produce blood group O. So basically, you see that the percentage chance of producing a child of blood group O, you see that if A is like this and B is like that, then automatically you're gonna have this, you're gonna be A, B, you're gonna have this, you're gonna be A, you're gonna have this is gonna be B, and then you're gonna have this, the small, it's gonna be small I, small I. So basically you have 1 out of 4 times 100, which gives me 25% chance. That's why I say that it looks to be 25% chance. What is the percentage chance of producing a, okay, we have seen it. What is the, we have, how many alleles are involved in the hybrid? How many alleles? So basically here, if you look at there are 2 characters which are being involved in this case. 2 what? That's why they call it a dihybrid cross. So each character will have 2 alleles. 2 alleles. So it means that there are going to be 4 alleles. So you guys, you have to take note of that. Yes, that in the dihybrid, we have seen it in our previous question. We have seen it in our previous question when we are talking about the first, I think it was the first question. You see that how many alleles? You have this, you have this, you have this, you have that. So there are 4 alleles which are involved in that case. But how many characters are involved? There are 2 characters. That's why you call it dihybrid cross. What is genetic engineering? When you talk about genetic engineering, it's you are engineering. We say that whenever I talk about engineering, you are manipulating. What are you manipulating? Genes. So manipulation of genes of organisms. Yes, this can be insertion, removal, what, transfer, all this. That's manipulation by the human, so that the organisms can produce a specific characteristics desired by the what? By the human. Yes, so that's biotechnology is part of genetic engineering. So give to advantage of genetic engineering. So we have seen this one before, and I don't think is now a big deal for that. So when you talk about genetic engineering, first of all, we say that medication, you can talk about pest control, you can talk about increase the yield, all this, increase the shelf life. Then the disadvantages: very expensive, interfering with nature, potential health impact, unsafe, unsure of the effects. All this, you see. Name the type of the what are the disadvantages? We have seen it. What are stem cells? These are undifferentiated cells. These are cells which are undifferentiated. They have the ability to divide and form, you see, undifferentiated cells that have ability to grow and form other or any tissue in the body. Yes. Examples is bone marrow, umbilical cord, tissue, adipose tissue, amniotic fluid. Yes, all these, they can be modified to form other body structures. So we call them stem cells. What are the sources of stem cells? What are the sources of stem cells? The sources of stem cells, we have seen them. What are the uses of stem cells? We see that they are very good in research. Very good in what? Research. New cells, we can do them for new cells. It's also good for replacing those cells which are damaged. Cancerous cells, all that are stem cells, they help us. So please don't go to the paper without knowing these stem cells. Then they're saying that what is cloning? What is cloning? This is the production of identical organisms. This is the production of identical organisms. How do we do that? We use what we call biotechnology. Then please make sure that you know I explained this and I don't want to repeat that too much. How do you produce this doll fish, the what? The sheep, dolly sheep. So when it comes to this, you have to get a sheep and you have to get another sheep. The sheep where the characteristics you want to produce, you get the udder. Udder is, or any other body cell. It must be diploid. You understand? You remove, because I want the characteristics of this sheep, so I keep the nucleus. And then this one, I have to get the ovum. Why ovum? So that you're able to get the casing. So I remove the nucleus because I don't want the characteristics of this sheep. And then after that, I bring this nucleus, I combine together using the mild electric shock. Then once I combine them together, I form something which looks like a zygote. It's not a zygote, but looks like a zygote because it is 2N, and the outside part is the ovum membrane. So that is it. So after that, we make it to insert it into this shape, the surrogate, and then this surrogate will have to undergo meiosis, then form different ball of cells, and then after that we get each pieces of this embryo development, and then we insert in different sheep. Each is going to become a sheep with the same genetic material as the original sheep. So that is it, and that's how it's being done. So why is it blood group not conclusive for parental test? Because there are so many individuals which have the same type of blood group. There are so many individuals who have the same type of bloody group. That's why. So here there are some questions. Those people who have distinction material book, yes, please use them. Then why is it not conclusive? Yes, it's not conclusive because of or because that many people have the same type of bloody group. So it means that you don't know which man is it who did what. Then you're saying that why is the male more affected by color blindness than female? Why are male more affected by color blindness than female? It's because males they have what we call they have one recessive, okay, they have one X chromosome, and then females have two X chromosome. So they require two recessive alleles for them to be affected, while male they only require one recessive allele for them to be affected. Female have two X chromosome, they require two recessive alleles for them to be affected by color blindness, while male have one X chromosome, they only require one recessive allele for them to be affected. Yes, so please go through these questions, make sure that you go through them. So it's time for evolution. So I will say that the paper is going to be very easy. It's going to be very easy. Make sure that you guys were able to perform and get a high mark when it comes to this. So the first question saying that describe the process of speciation through geographical isolation. Describe the process of speciation through geographical isolation. So it means that you have to describe how new species, because speciation means formation of new species, but using a barrier. That's what you call the geographical isolation. So in this case, this question is just open. It is generic. It does not specify which barrier. So you describe the process of speciation. So you explained that there was one population of organisms. This population are being separated by a river, or let me just use also the distinction material book. By a river, it could be a river, it could be a mountain, and then this population then split into two. Yes, after splitting into two, then you explain that there is no gene flow between these two populations. Each population may be exposed to different environmental conditions. Therefore, natural selection occurs differently, and then these organisms, they became genetically different and then phenotypically different. And then when you remove the barrier and then let them mix, they cannot interbreed to produce a fertile offspring, and then now they have become new species. So that is what you're supposed to explain. So if we look at it here in our distinction material book, we see that this is the speciation you're supposed to talk about. Talk about this there. Yeah, there's one population of a particular species is a generic like this, exactly like this, split into two by geographical barrier, river, mountain, whatever, then resulting into two parts of this population. You understand? And then there is no gene flow between these two populations. The natural selection occurred independently, and then due to different environmental condition, yes, the two populations became genotypically and then phenotypically different. Even if these two populations mix, again, they cannot interbreed to produce a fertile offspring. Therefore, one or both of them have become new species. So that's how you explain. But if they give you an example like this, or like this, or like this, you have to describe according to the example. So here there's one population of rabbits. The population was divided into two by the river, into population A and B, and then it means that there is no gene flow between these two populations. So natural selection occurs independently, and then if you so since it occurred independently, they became genotypically and phenotypically different. Yes, if you remove the river and then you mix them again, they cannot interbreed. They cannot interbreed to produce a fertile offspring. Therefore, they have become new species. Species X and species Y have become new species of rabbits. So you have to use the example they have given you to describe that concept. Then they are saying that this one question is about human evolution. The diagram below represents the skull of hominids. List the three similarities in relation to vision that are shared. If you look at them, eyes are in front. Yes, they have two eyes. All of them, they have two eyes, and then they're able to detect color, and then that's why we say that they say there are three. They have two eyes in front. They're able to detect what? Color. So those are the three maybe characteristics you can talk about related to vision. They say related to vision. So when they are giving you these questions, sometimes they are specific to that specific part of it. Maybe the hand, maybe the teeth, maybe so it will depend on what specific part they want to focus on. Name one species in the diagram that was prognathous. That was most prognathous, the one which has the protruding jaws. So it is gorilla is the one which has more protruding jaws. If you check here, that it's protruding out much more compared to this one is too much compared to this and this. So it means that is gorilla. That's the answer. How did the Homo describe two structural features, two structures that caused the organisms named above to be most prognathous? First of all, if you look at it, we see that if you look at it, it has the protruding jaws. The jaws are large. If you look at them, the jaws are larger compared to this. And also, if you look at, let's look at the canines, and then we see. If you look at its canine, we see that its canines are big. So this canine require enough space. That's why they have protruded out. So large canine or large teeth, and then also the large jaws. So they become more prognathous. All right, Homo naledi was bipedal for most of its adult time. Explain how the structure of Homo naledi's skull would have assisted in bipedalism. Homo naledi, if you look at it, first of all, the position of foramen magnum is in a more forward position. A more forward position of foramen magnum, which allow the spine to connect vertically, to support the what? The skull. So it is in a more forward position. If you look at it here, it is in a more forward position. It allows the spine to connect vertically so that you are able to carry this skull into the sky or in the space. So explain how the Homo naledi skull would have assisted in bipedalism. Remember this one, we said that it is the ability to walk on two limbs. So anything related to this is related to the position of the foramen magnum. If it is in more forward position, you must use the word more forward position. If you don't use the word more, then it means that your answer is going to be wrong. So the foramen magnum is in a more forward position to allow the spine to connect vertically so that you are able to support the skull in the what? In the space. The next question is describing the difference between So we are saying that describe the difference between Homo sapiens and the gorilla in relation to the spine. If you look at the Homo sapiens, this one is bipedal and then this one is quadrupedal. So it means that if you look at the gorilla, it is C shape, while the humans, it is S shape. So gorilla, we see that Homo sapien has an S-shaped spine, and then gorilla has a sheath-shaped spine. That's number one. Then if you look at Homo sapiens, if they ask you for the pelvis, is short and wider, while the gorilla is long and narrow. If you check it on our distinction material book, these things we stipulated them properly. So short and wider, while the other one is long and narrow. Yes, this is the gorilla, and then that is it. So the spine, we said, is short and wider for the case of human body, and then for the case of the gorilla is long and narrow. While the spine is S-shaped spine, and then this one is a C-shaped spine. Then we go to the next question. They are saying that explain why the gorilla species has cranial ridges. It depends on what it was feeding on. Because the gorilla, if it was feeding on raw tough food, therefore it requires muscles for it to be able to to chew that food. So basically those ridges are responsible for muscles. So you can say that for muscle attachments, so that these muscles are able to assist during eating tough or rough or strong food. So for muscle attachments, muscles, they must be strong enough to be able to chew this food during, remember the food is eating is raw. So this cranial ridges is for muscle attachment to assist this gorilla in eating this raw food. Then they're saying that you have the skulls and the pelvis. State the name the genus for A. sediba. That is Australopithecus. Sediba is Australopithecus. A means that it is Australia. Austral. You see Australia. You see Australopithecus. Australopithecus. Yes, Australopithecus. Sediba, it means that that's the genus. First name is the genus, the second name is the species. Describe the shape of the spine for Homo sapiens. We say that it's S-shaped spine. S-shaped spine. Then they're saying that Australopithecus sediba is thought to be transitional fossil. What is a transitional fossil? Remember we said that transitional fossil is a species which has both characteristics of the ancestor and that one of the what? Of the ancestor and the immediate or the one which follows it. Is the one which shows the intermediate characteristics between two genera, or is the one which with the characteristics of both ancestor and the species that follows it. So organism that has intermediate or common characteristics between two genera, so that is a transitional species. Then we go to the next question. Give the letter of the previous of the pelvis that would be representative of Australopithecus sediba. If you come back here, you of what? Of the pelvis. Yeah, it's not this. This is gorilla. Then this is human, and then this is Australopithecus sediba. So the answer here is gonna have to be A. State what is meant by transition of. Oh, we are done with that. No. Give the letter. The A is here. They are saying that explain your answer because the pelvis. If you look at the pelvis here, the pelvis that is intermediate is transitional. This pelvis, it has some characteristics of this and has some characteristics of this. So remember, Australopithecus sediba is a transitional fossil. We said it. Australopithecus sediba is a transitional fossil or species. It means that it has characteristics of both ancestor and the species that follows it. Then we go to the next question. What is the significance of change in the prognathes? Significance, explain the significance of change in the prognathes from Australopithecus sediba to Homo sapien. So Australopithecus sediba was prognathes, meaning that it was prognathes, while humans was non-prognathes. You understand? Yes. This is due to the smaller jaws. Yes, it's due to the what? The smaller jaws. These jaws, it made them to have small teeth to reduce the chewing what? Muscles caused by the change in the diet to eat the soft cooked what? Cooked food. So the significance in the change in the prognathes, in other words, from sediba to Homo sapiens, is because of the change in the diet, and that change, it made it to have to be non-prognathes, meaning that it does not have muscles where these jaws are supposed to be attached. The muscles on the big jaws and the ridges, so that those muscles can be attached for raw meat. So, in other words, when you change from soft, rough food to soft food, also it's like when you are living a tough life and then you change to a soft life, even the way you think gonna change, because everything's there for you, they are thinking for you, everything, so you can't even reason properly. You understand? Yes. Why? But that comes after many generations. It doesn't happen on you. It will happen in other generation if you don't become careful with that kind of lifestyle. So if it was feeding on raw food, that's Australopithecus sediba, and then now this one is cooked food, automatically it's gonna become changing from prognathes to non-prognathes. It's gonna change from large jaws to small jaws. It's gonna change from big canines to small canines, and then that it means that it's gonna have to reduce the chewing muscles. Yes, from big ones to small ones due to the change in the diet. So that's what it means. The table below shows the information of selected hominid species. So we have the hominid species here in the table. So we're going to have to answer those questions. So if you look at it, Australopithecus sediba, we call it carabo. And then you have Australopithecus africanus. yes, which is Taomchild, Australopithecus africanus, Taomchild, Mrs. Place, Littlefoot. That's Australopithecus africanus. And then where do we discover Karabo? Karabo was who discovered Karabo? That is Lee the Beggar. Lee Beggar. Yes, and then who discovered Taomchild? That is Raymond Dart. Raymond Dart. Name the P. We are done with that. P. What is P? We are done with that. Identify the species. R. Name the scientist. So we have got all that information. Then they are saying that this is the phylogenetic tree. Identify the type of diagram shown. Identify the type of diagram shown above. The type of diagram shown above is called a phylogenetic tree. That is a what? A phylogenetic tree. So when it comes to the phylogenetic tree, you need to understand some few concepts. You need to know how to calculate it. You need to explain it. So the diagram below shows, we're saying that genus, give the genus, identify the phylogenetic tree. Then they give the genus that rise from Australopithecus africanus. Australopithecus africanus, Australopithecus africanus. Where is it? Australopithecus africanus. The common ancestor, all of them, Australopithecus africanus, they come from the genus that gave rise to Australopithecus africanus. Australopithecus africanus is here. So basically it's supposed to be Adipithecus. 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 Remember, it is Adipithecus remedus which produced the Australopithecus africanus. You see here? Yeah. It is Adipithecus remedus which gave to Australopithecus afarensis and then Australopithecus africanus. But they are looking for the genus, so it means that is Adipithecus. Then they're asking you the letter that represents the most recent ancestor for Homo sapiens. The most recent ancestor for Homo sapiens. Homo sapiens. Yes, if you look at the Homo sapiens, they are here. This is the most recent ancestor, so it's going to be C. And then they are saying that name two species that existed at the same time as Homo erectus. Same time as Homo erectus. Where is Homo erectus? Homo erectus is here. So I'm going to have to draw a line. So we see that gorilla existed. And then also troglodyte also existed, and then the recent the ancestors of Neanderthalensis, Homo neanderthalensis and Homo sapien. You see, yeah, there. So all of them, they exist at the same time. Then they are saying that name two species that, okay, we're done with that. Homo erectus. Give one example of fossil of Australopithecus africanus that existed in Africa, in South Africa. That is Taomchild, Mrs. Place, and then Littlefoot. Yes. And then they're saying that evidence. We go to the evidence, state the out of Africa hypothesis. When you look at the modern humans originated from Africa and then migrated the rest of the world. Modern humans originated from Africa and then migrated to the rest of the world, or to the other parts of the world. So that is it. Describe how fossil evidence is used to explain. Guys, whenever I told you that when it comes to fossil evidence, you tell us the oldest fossil, which one is the oldest fossil, and then you tell us that fossil evidence, Adipithecus were only found only in Africa. The word only must be there. Fossil of Australopithecus, Karabo, Australopithecus was Karabo, Littlefoot, Mrs. Place, and Taum Child, yes, were found in Africa. Homo habilis were only found in Africa. Only all these Homo habilis, Australopithecus, Africa. Then Homo erectus and Homo sapien, then we use the word oldest. So this word only, and here only, is very crucial, while this one is the oldest, then for Homo sapiens and Homo erectus. So you take a screenshot, make sure that you know that before you go to the paper. Then the next question, they're saying that the table below shows the timeline of different hominid species and the development of tools. So they're asking you which species in the diagram above existed or survived for the longest period of time. If you look at this, we shall see that it is Homo erectus which survived for the longest, because this shows the time it existed. Then they are saying that calculate the period in million years in which Australopithecus afarensis and Africanus coexisted. Afarensis, Australopithecus afarensis. This is afarensis and Africanus. So it means that it started from here and ended here. You see? That's the time when they coexisted. So I'm going to come here and read. Remember, I read from 0. So I'm reading from this side. So it's around 3.2.4, 3.4. I hope it's 3.0. It's each is 1, 1. 3.1, 3.2, 4, 5, 6, 7, 8, 9. Yeah, 3.2. So it's going to be 3.2 minus. Then here, if it is 1, 2, 3, 4, 5, 6, 7 minus 2.7, the answer I get is going to be in million years, which is 0.5 million years. So don't forget that you count from the 0 present. You don't count from this, because now you are used to count it from there. Then they're saying that name the species that was also known as Handeman. That is the Homo habilis. Homo habilis. Then they're saying that name 2 species that use tools, that developed tools. That is the state 2 tools. Okay, we say that tools were used, tools. Yeah, 2 species that use tools. So when you come back here, we see that state 2 uses of tools. Oh, what did they use? Check here. They say that complex tool arrow used to stick and fish. Okay. Then surface here for digging and axe. Okay. Scrapping. What did they say here? Name 2 uses of tools that were developed. 2.5, 2.6 billion. Oh, is here. So it's these tools, which is for scrapping, pounding, and chopping. Yes, so it means that they only wanted 2 answers there. Please, when it comes to this, you have to be applicable in that thing. Then they're saying explain how the change in the brain size over time relate to the development of tools. Increase in the brain size led to increase in the intelligence, leading to the development of complex tools. As the brain size increase, there will be more intelligence. The intelligence increased, and then leading to the more complex tools. So that makes us to go to the catch-ups. You guys, catch-ups is simple. Differentiate between the hypothesis and the theory. A hypothesis, the tentative statement which is subject to be proven correct or wrong, while theory, it must be based, it must be supported with the facts, it must be supported with evidences, tested hypothesis also, and others. Modification by descent, that is homologous structures. That is modification by descent. Continuous and discontinuous variation. Here, guys, you have to look at what you were born with, and it keeps on changing, like skin color, size of the shoes. the length of the hair, all that is continuous. Where this continuous blood group, you can check on our distinction material book. Blood group, you can talk about tongue rolling, which are genetical, too much controlled by genetics. So those are the examples. State Lamarck's law, it has a law of use and disuse. The more frequently you use the structure, the more it becomes better adapted. And if you don't use it, it becomes smaller and finally disappears. Then why is Lamarck's law rejected? Make sure that you apply. It was rejected because of what? It was rejected because organisms don't evolve because they want to evolve. No, organisms don't evolve because they don't want to evolve. Yes, and then there is not enough evidence to support that the characteristics which are obtained during lifetime are being passed to the next generation. Because the second law says that the characteristics we obtain during lifetime are being passed to the next generation. You see? Yes. So in this case, you have to be specific which characteristics are they talking about. Give two difference between Lamarck's theory and Darwin's theory. You come back here and then you look at Lamarckism and Darwinism. Variation among of offspring occurs when individual in a population. Well, here the whole offspring. Variation occurs in the offspring. The variation basically you obtain from your offspring. Here I talk about only one individual will change. Here the whole population changes. So at least make sure that you know at least 2 points in that regard. Then they are saying that punctuated equilibrium and gradualism. We said that in punctuated equilibrium, organisms gradually change. Here the organism gradually change. While in punctuated equilibrium, there is short period of rapid change and long period of no change. So the change is just drastic. Turned from this to this, turned from this to this. While here it keeps on changing. There is intermediate in between. Here there are no intermediates. You see? Yes. Then we say that what is artificial selection different from natural selection? Basically, artificial selection is different from natural selection due to, number one, natural selection, nature is a selective force. Artificial selection, human is the selective force. You see? Selection is in response to the suitability of the environment. Here, it's a response to the suitability of the human needs. So at least know two, and then you continue with your life. Then you're saying that describe how selective breeding is being done. We say that here in selective breeding, what you do is you look at the organism which is needed to be obtained. For example, milk production, drought resistance. So what do? Get the one which is you want to the desirable characteristics. For example, choose the cow which you want in the herd which can produce more milk. You understand? Let those cows which can able to have that characteristic to reproduce, and then after that give those cows, find out which one is producing most milk, and then let them reproduce again. You keep repeating that until you get your desirable what goals. So that is selective breeding. You have to know that. Then we are saying that describe the selective breeding occurs in cows. What is speciation? We said the formation of new species. What is population? Is a group of organisms of the same species able to interbreed, produce fertile offspring. They are living together in a specific time. Yes, at a specific area. Species, the group of organisms, they have the same characteristics, able to interbreed to produce fertile offspring. Describe how new species are formed using geographical barrier. We talked about it. Antibiotics, and then describe how evolution of natural selection. So I'm going to talk about this, and I'm going to talk about this, and then you go to human evolution. So when it comes to evolution by natural selection, you talk about that organisms produce a large number of offspring, but these organisms, like Lamarck's theory, how did they Darwin's theory, how did they have to be with this. Organism produce large number of offsprings. You tell us that there is a great deal of variation, like giraffes produce there is a variation in the giraffes, those which are long necks and those who are short necks. You are busy describing natural selection. You understand? Those which have long necks, they are able to compete and survive in the environment. Why? Because they're able to reach the tall tree plants, so that they're able to get the food. While those with the short necks, they're not able to get the tall tree plant to get food, so they die out. So they have unfavorable characteristic. Those with long necks, they pass their gene to next generation. Then in many generation, all offsprings have long necks. So that is evolution by natural selection. Then the present evolution, you talk about this mosquitoes thing, that there is variation, still the same thing, variation among mosquitoes. And when you apply the DDT or insecticide or antibiotics, those which are non-resistant, because there is variation among these mosquitoes, those which are resistant to DDT and those which are not resistant. Those which are resistant, they will survive, while those who are not resistant, they die. Those which are resistant, they produce in large numbers. So the next generations will have more resistant mosquitoes than the non-resistant. So that now, if you apply this DDT, it won't be effective. It will be ineffective, meaning that now the population of mosquitoes will have more resistant mosquitoes than the non-resistant. Then human evolution. Which family do we belong? We belong to Hominidae. Humans belong to family Hominidae. That is Hominidae. Then they're saying that give two differences between humans and other primates. Here it depends. We answered it, but it depends on what they have asked you. So in this case, you look at the upright posture, large brain, eyes in front, all those answers. Tabulate the difference between the cranial capacity of human and apes. Cranial capacity of a human is large to accommodate more brain cells, high intelligence, while this one is small. That's why the intelligence is a bit smaller. Tabulate the difference between humans and the other primates in terms of the skull. So this is what you're supposed to do, these ones. And then this table is here from our distinction material book. Large cranium, small cranium, flat face, sloping face. But in most cases, we don't bring you like this. We bring you the same way we brought it here, and then we started to ask you questions about this. So but you have to know that this means this means this. Prognanthus, non-prognanthus. Prognanthus, non-prognanthus. For the case of human, this is prognanthus. Remember, we modified that. Please make sure that you are able to get it right. 3 factors that lead to bipedalism: shape of the spine, pelvic girdle, position of foramen magnum. Advantages: we say that you are able to, your body is lifted from the ground. You're able to see far. You're able to carry because the hands are free, you're able to carry maybe weapons and stuff. You see, movement becomes easier. So advantages of bipedalism. Give 2 differences with evidence other than the fossil evidence. Yes, we have the mitochondrial DNA, and then we have what you call a cultural evidence. Don't say use of tools. Cultural evidence is tools. Mitochondrial DNA, use of the mitochondria. What is the transition fossil? We said this is a fossil which has both characteristics between 2 genera. Yes, example is Australopithecus sediba and Homo naledi. Give 2 Australopithecus africanus species found in Africa. We said Taon child, little foot, and then Mrs. Place. Which organism used first to use the tools? Homo habilis. Which organism first walk on 2 limbs? Homo erectus. Who discovered Australopithecus sediba? You have to know the whole page, and we say that who discovered Australopithecus sediba? The answer was very clear, and we say that Australopithecus sediba was done by Lee de Berger. Yes. So you see, you have to know this table, guys. You have to know this table. Then they're saying that what is the common name for Adipithecus remedus? Common name is Ad, is Mr. Ad. Yes, add. How is dentition they show the line of evolution? We say that dentition is large canines, shows that you are feeding on raw tough food. Yes. And then if you have small canine, then it means that you are feeding on soft cooked food. Then the last question which you need to do is this question. Please make sure that you go through it, and I think take a screenshot of it, ask questions, then we'll be able to answer you. Please don't forget to check this book from our link just by clicking join. Then we'll be able to get that book so that it's able to help you and prepare for your exams. Guys, I wish you the best. It has been M. Saidi, and I know that you're gonna make it. All the best, and don't forget to like, subscribe, and share for our daily new videos.
Welcome to Thundereduc, an online learning platform powered by professional, highly skilled and qualified educators.
We deliver structured CAPS online lessons for high school subjects including mathematics, physical sciences, life sciences, geography, business studies, history, accounting, English, Afrikaans, and others.
For the intermediate and senior phases, we offer expert teaching in natural sciences, mathematics, social sciences, English, and EMS, designed to build strong academic foundations.
Access the distinction material books by clicking join button on our YouTube channel, or download Thundereduc app on Google Play Store for all our study material and past exam papers.
Thundereduc offers extra classes for rewrites, old curriculum, and other students from grade 4 to grade 12 through online and physical classes.
Subscribe to our YouTube channel, Thundereduc, for reliable curriculum-focused learning and stay updated with the latest educational content.
Please like, share, and turn on notifications so you never miss a lesson.
Visit us at www.thundereduc.com
or call 0786 636 613 or 084 833 1738 for more details.
Thundereduc, we focus on quality.
Welcome back to our class. Today we're going to be talking about the prediction for paper 2. So basically it could be a prediction or a scope, but let it be a guideline for you to prepare for your exam. And I know that by the end of this lesson you must be able to get a distinction. Those who have the distinction material book, I know that the distinction is already in your hands. All right, let's start without wasting time. The first question. The bond that forms between the amino acids during protein synthesis. How do you call that bond? So we call that bond peptide